Welcome back to the Deep Dive. This is where we take complex, expert sources and really break them down into the knowledge you need, helping you get up to speed quickly, but also deeply, like the top professionals in the field.
That's the goal.
And today, we're tackling a huge one, the incredibly intricate, constantly changing world of infectious diseases. Okay, let's unpack this.
Right.
We're basing this entire deep dive on gold standard material, specifically digging into the core principles laid out in Davidson's Principles and Practice of Medicine.
A foundational text.
Absolutely. And we're not just listing facts here. We want you to grasp the fundamental why and how behind infections. Everything from the tiniest microbes causing disease
All the way up to global prevention strategies and that really complex dance of antimicrobial therapy.
Exactly. We're aiming to cover it all so you can really master this information, building that elite professional level of understanding.
That's right. Our mission today is exactly that. To get you to a point where you truly understand the biological principles, the epidemiology, the diagnosis, prevention, treatment, all of it.
Not just the what, but the why.
Precisely. The kind of deep insight that medical professionals rely on every single day. And what's fascinating here, and maybe a little unsettling, is that this whole field is never static, is it?
No, constantly shifting.
Microorganisms are always mutating, always evolving. So new infectious agents, new forms of antimicrobial resistance. Well, that's just inevitable, frankly.
Which makes this foundational knowledge absolutely critical.
It underpins everything else. It gives you the tools, the critical thinking to navigate what's coming next.
Okay, so let's start right at the beginning, the fundamental question. What is an infectious agent? We have to go back to the roots of microbiology here.
And that really means going back to Robert Koch in the 19th century. He was absolutely pivotal in establishing this core concept.
He basically formalized it, didn't he, with his postulates?
Exactly. Koch's postulates, they were revolutionary for their time, a systematic way to link a specific microbe to a specific disease, moving beyond just saying they seem related to actually proving causation.
And they're still taught today, which shows how important they were. So there are 4 key steps he laid out.
Right.
First, the organism has to be present in every case of the disease.
Right.
No exceptions.
Okay.
Second, you have to isolate that organism from the diseased host and grow it in a pure culture in the lab.
Right. Get it on its own.
Third, you take that pure culture and inoculate a healthy, susceptible animal. And it has to cause the same disease.
The crucial test.
And finally, the 4th step. You have to be able to re-isolate the same organism from that newly diseased animal. It's a pretty rigorous chain of evidence.
It was incredibly rigorous. But, it's also really important to understand the limitations. Koch's postulates, well, they don't work perfectly for everything.
Ah, okay. Like what?
Well, think about organisms you just can't culture in the lab using standard methods, like Mycobacterium leprae, the cause of leprosy, or Trophorema whipplei for Whipple's disease. You just can't grow them easily, so that second postulate.
You can't fulfill it.
Exactly. And then there's the issue with organisms that are part of our normal human flora, bugs like E. coli or Candida species.
Things that are usually harmless.
Right. They live on us or in us quite happily most of the time. But under certain conditions, they can cause disease. So the first postulate, being present in every case, gets complicated. And sometimes they're always present, even in health.
So it raises that question. If you can't culture it, or if it's always there, how do you definitively pin the blame on that specific organism?
Precisely. It shows why we need more than just Koch's postulates today. We need molecular methods, immunological evidence, a broader diagnostic toolkit. It highlights the complexity right from the start.
That's a really critical point. And speaking of definitions, let's nail down some terms that often get mixed up. Infection versus disease.
Yes, good idea.
So strictly speaking, infection means the microorganisms or other agents have actually established themselves in the host cells or tissues. They're replicating, causing some harm, and triggering a response from the host.
Okay, establish and causing trouble.
Right. Now, if they're just surviving and multiplying on a surface like your skin or mucous membrane, but without causing harm or illness, that's called colonization.
Just hanging out, basically.
Pretty much. Then there's latent infection. This is where the microorganism invades, but then it just lies dormant, survives inside cells or tissues without causing active symptoms. Still there, but quiet.
Like TB can do, for example.
Exactly. And finally, infectious disease. That's when the agent itself, or maybe the host's response to it, is enough to actually cause illness or harm. Most disease-causing agents, or pathogens, are microorganisms, but some are multicellular, like parasitic worms.
And it's interesting, sometimes the symptoms of the disease, like, say, diarrhea, actually help the pathogen spread to new hosts. A rather grim evolutionary strategy.
Grim but effective.
And building on that distinction, while we often use infection and infectious disease almost interchangeably, it's worth noting that not all infections are technically infectious in the sense of being easily passed from person to person. We usually call diseases transmitted between hosts communicable diseases, but then you have endogenous diseases, which are caused by organisms that were already colonizing the host, your own flora turning against you, essentially.
Like we mentioned with E. coli causing a UTI sometimes.
Exactly. But the lines can get really blurry, can't they? Especially with healthcare-associated infections, HCAIs. Think about MRSA or C. difficile.
Right.
Colonization often comes first. The bug is just sitting there. But then the infection develops. And the tricky part is that colonizing bacteria might have actually been transmitted from another patient recently.
So it's endogenous and potentially communicable.
Kind of both, yeah. Which is why understanding the whole chain of infection is so incredibly important. It maps out the entire journey.
Okay, let's look at that chain. It's a really useful model, often shown as a diagram. Think of it as the 6 essential steps needed for a communicable disease to spread. You need all 6 links.
Break one link, you stop the spread.
Exactly. So first, you need the infectious agent, the bug itself. Second, the reservoir, which is where that bug population normally lives and multiplies. Could be a person, an animal, maybe the environment.
The source.
Right. Third, the portal of exit. How does the agent get out of the reservoir? Coughing, sneezing, feces, blood.
Okay.
Fourth is transmission. How does it travel from the reservoir to the next person? Direct contact, airborne droplets, contaminated food or water, maybe an insect vector like a mosquito, or even an inanimate object, a somite like a contaminated surface.
Lots of ways.
Yep. Fifth, the portal of entry. Where does the agent get into the new host? Respiratory tract, mouth, broken skin, mucous membranes.
The way in.
And finally, number 6, you need a susceptible host. Someone whose body can actually be invaded and get sick from that agent. If you're immune, that link is broken.
And what's fascinating here, as you said, is how understanding every single one of those links is the absolute key for prevention and control. Because public health interventions are all about breaking that chain somewhere, it raises that crucial question for anyone in the field. Where's the most effective place or places to intervene?
Is it vaccination, targeting the host?
Or sanitation, hitting the reservoir or transmission route? Maybe isolation, focusing on the portal of exit?
Or even vector control, like mosquito nets.
Exactly. It's often a multi-pronged attack, hitting several links at once. It's strategic.
Okay, so we've covered the what's and the how of spread. Now let's dive into the actual agents themselves. There's incredible diversity here.
A whole rogues gallery.
Starting with viruses. These guys are the ultimate biological hijackers, aren't they?
Absolutely. They can't replicate on their own. They have to get inside a host cell. They're obligate intracellular parasites.
Meaning they completely take over our cellular machinery to make copies of themselves, their nucleic acids, their proteins.
And their genetic material can be DNA or RNA, single or double-stranded. Then you have retroviruses like HIV.
Which have that extra step, right? They use reverse transcriptase to turn their RNA into DNA inside our cells.
A very clever trick. Structurally, you've got the genome wrapped in a protein coat, the capsid. That forms the nucleocapsid.
And many have an outer lipid envelope too.
Right, and that structure really matters for survival. Enveloped viruses, often the ones spread via respiratory, sexual, blood routes, or insects, tend to be less stable in the environment.
More fragile.
Yeah. While the non-enveloped ones, often spread fecal-orally or sometimes respiratorily, they tend to survive better outside the host.
Which influences how we control them. Our source shows the generic life cycle too. Attach, penetrate, uncode, replicate, assemble, release, either budding off or just bursting the
It's
stack.
a hostile takeover at the molecular level. And just a side note, viruses that infect bacteria are called bacteriophages or phages. A whole other fascinating world.
Okay, moving from viruses to prokaryotes, primarily bacteria, but this group also includes things like mycobacteria and actinomycetes.
And the big difference from viruses is that these guys can reproduce on their own. They can make their own proteins, their own nucleic acids. They just lack a nucleus, unlike our cells.
And they have that characteristic cell wall made of peptidoglycan.
Crucial structure. And it varies. Thick in gram-positive bacteria, much thinner in gram-negatives.
And gram-negatives have that extra outer membrane containing lipopolysaccharide, LPS.
Which acts as an endotoxin, A major trigger for our immune system, often causing fever and inflammation.
Right. Inside, they have their main chromosome, but also often those extra little rings of DNA.
Plasmids. Really important. They can carry genes for things like antibiotic resistance and can be passed between bacteria quite easily.
Spreading resistance around. They might also have a capsule for protection or flagella for moving around. And while most bacteria can live independently, some are like viruses in that they have to live inside our cells. Obligate intracellular bacteria like chlamydia or coxiella.
And this is where the deep dive gets really interesting for treatment. You mentioned the cell wall difference, gram-positive versus gram-negative.
Yeah, thick versus thin, peptidoglycan layer, and the outer membrane on gram-negatives.
That structural difference is absolutely fundamental to how many antibiotics work. Beta-lactams, like penicillin, they target that peptidoglycan synthesis.
So they're generally better against gram-positives with their thick, exposed wall.
Much more effective, generally speaking. The outer membrane of gram-negatives acts like an extra barrier, making them naturally less susceptible to some drugs or requiring different types of antibiotics that can get through it.
So that simple gram stain test in the lab.
It's one of the first, most critical pieces of information you need to even start thinking about which antibiotic might work. It's basic, but absolutely essential for guiding therapy.
That leads perfectly into how we actually identify these bacteria. It's like microbial forensics.
Creating a fingerprint for the culprit.
So first, the gram stain reaction, positive, purple, negative, pink, or maybe unstainable. Then microscopic morphology. Are they cocky spheres or bacilli rods? Do they have a capsule?
How they group together matters too, right? Clusters versus chains.
Exactly. Staph in clusters, strep often in chains. Then you look at their colonial characteristics on an agar, plate size, shape, color, maybe hemolysis, that clearing or greening on blood, agar.
Beta or alpha hemolysis.
Right. Their atmospheric requirements are key too. Aerobic, anaerobic, facultative, microaerophilic. What kind of air do they like?
Or dislike.
The biochemical reactions, what enzymes do they have? What can they ferment or break down? Are they motile? And crucially, inherent antibiotic susceptibility patterns can be a clue.
Like strep pneumonia being sensitive to optogen.
That's it. But beyond these classic methods, we now have amazing rapid techniques.
Melitov mass spectrometry is a big one.
Yeah, Melitov MS. It identifies bacteria, even some fungi, super fast based on their unique protein profile, like a molecular fingerprint.
Incredible technology, and 16's ribosomal RNA gene sequencing.
Very specific for identifying bacteria, especially from sterile sites or pure cultures.
Yeah.
And looking forward.
Whole genome sequencing, WGS. It's not quite routine everywhere yet, but the potential is enormous.
To get identification, sensitivity, and typing, maybe directly from the patient sample.
That's the promise. It could genuinely revolutionize diagnostic microbiology in the near future. Replace a whole suite of current tests with one powerful analysis.
Wow. Okay, so from prokaryotes, let's move to the eukaryotes. Fungi, protozoa, and helminths. These are more complex cells, like ours, with nuclei and other organelles.
Right. Fungi come as molds, filamentous, or yeasts, single-celled. Some are dimorphic, can switch between forms.
And a key difference from our cells, which is great for drug targeting, is ergosterol in their cell membranes instead of cholesterol.
And they have a cell wall, different from bacteria, made of polysaccharides, chitin. Beta-glucan is a major component.
Again, targets for antifungal drugs.
Exactly. Highlighting these structural differences is crucial because, just like with bacteria, they give us specific targets for drug development. Ergosterol synthesis inhibitors, beta-glucan synthesis inhibitors. These allow us to attack the fungus while minimizing harm to human cells. It's about exploiting those biochemical differences.
Smart. Then you have protozoa and helminth, often just called parasites. Many have really complex life cycles, sometimes involving multiple different hosts, which makes controlling them extra challenging.
Definitely.
And finally, just a quick mention of prions. They're transmissible, infectious agents, but they're not microorganisms. They're misfolded proteins.
So they're diagnosed differently, not typically in a standard microbiology lab, a unique category.
Okay, shifting focus now from the outside invaders to the world inside us, the human microbiota. It's staggering how much life we carry around with us.
Trillions of microbes.
We're talking about the normal microbial flora.
Yeah.
These are the microorganisms that live on our skin, in our mouth, or gut. surviving and replicating, usually without causing any harm. They're colonizers.
And the gut and mouth are the most densely populated areas, each with its own distinct community doing different jobs.
Understanding this normal flora is vital for doctors
Yeah.
for taking samples and interpreting results.
Yeah.
You don't want to mistake a normal resident for a pathogen.
Absolutely. Knowing what should be there is key to spotting what shouldn't, or what's out of balance. Figure 6.5 gives a great overview of this diversity across body sites.
And this collection of microbes, their genes, how they interact with us in the environment, that's the microbiome.
And we're learning more and more about its profound influence on our health and also on disease. It's not just passive colonization.
No, these microbes are actively beneficial. They synthesize vitamins for us, like K and B12 in the gut.
And they provide colonization resistance. They actively prevent more dangerous pathogens from getting a foothold.
How do they do that?
Several ways. They can change the local environment, like making it more acidic. They produce their own antibacterial compounds, like bacteriocins. And they even stimulate our immune system in ways that can help protect us against related pathogens.
So it's this incredibly delicate balance. Maintaining this internal ecosystem is absolutely vital for our health.
It's a true symbiosis, a constant negotiation and collaboration happening inside us all the time.
But while places like our gut are teeming, other parts of our body need to stay sterile. How does the body manage that? How does it maintain sterility?
We have some clever physical barriers and mechanisms. The mucociliary escalator in our airways, for instance.
Like a constant conveyor belt, moving mucus and trapped microbes upwards.
Exactly, sweeping things out. In the urinary tract, the urethral sphincter acts as a barrier preventing bacteria from traveling up into the bladder.
And then the obvious ones, skin, the lining of the gut, mucous membranes generally.
They form a crucial physical wall, keeping the underlying tissues, bloodstream, and body cavities sterile. Essential fortifications.
But sometimes, despite these defenses, our normal flora goes rogue. It causes endogenous disease.
Right. This happens mainly in a couple of ways. First, translocation. The normal bugs spread from where they belong to a normally sterile site.
Like gut bacteria causing a UTI, or skin bacteria infecting a surgical wound.
Perfect examples. The other way is overgrowth. The organism stays in its normal place but grows excessively, upsetting the balance. Think dental cavities, vaginal thrush, or blind loop syndrome in the gut.
Where bacteria overgrow in a part of the small intestine.
Exactly. And then there's cross-infection, where harmless flora from one person gets transferred to someone more susceptible and causes disease there. Often happens in hospitals.
It really highlights why there's growing recognition of the need to limit how much we disrupt the microbiota with antibiotics.
Definitely. Wiping out beneficial flora can open the door for problems, which leads to the idea of probiotics.
Giving beneficial microbes back to try and restore balance.
That's the concept, giving specific microbes or mixtures to prevent or treat infection. But, the evidence for their clinical benefit, it's still evolving. There's a lot of debate and ongoing research. It's not a simple magic bullet.
Okay, so from the normal balance, let's turn to the conflict. Host-pathogen interactions.
Yeah.
The battle.
Right. We talk about pathogenicity, the ability of an organism to cause disease, and virulence, the degree or extent to which it causes disease.
And pathogens achieve this using virulence factors, specific molecules or proteins that help them cause harm. They're weapons.
And we can broadly categorize pathogens into primary and opportunistic.
Primary pathogens can cause disease in healthy people.
Yes, if you're exposed, a certain proportion of people will get sick regardless of their immune status. Opportunistic pathogens, though, they only cause disease when the host's defenses are weakened.
Like if someone has an IV line or is on immunosuppressant drugs?
Exactly. The opportunity is the compromised host defense. It's a key clinical distinction.
So what makes a pathogen successful? What are their characteristics?
They have quite an arsenal. Competition is key, stealing nutrients, producing those bacteriocins to inhibit rivals.
Motility helps them reach places other bacteria can't.
Right, like deep into the lungs.
Yeah.
Adhesins are crucial molecules that let them stick to our cells, often the very first step.
Gaining that foothold. And some can directly invade tissues too.
And then there are biofilms. These are a major problem.
That's where bacteria form these complex 3D communities.
Exactly. They adhere to a surface, multiply, and then secrete this protective matrix around themselves made of host stuff and bacterial products, like building a fortress.
Which makes them incredibly hard to treat, especially on medical devices.
Precisely. This raises that important question. Why are they so difficult to treat? It's because that matrix shield and the altered state of the bacteria inside makes it really hard for antibiotics to penetrate and work effectively.
So often you have to remove the device entirely.
Often, yes. That's the only definitive way. It shows how formidable biofilms are.
Pathogens also deploy toxins, microbial molecules causing damage.
Two main types, endotoxin and exotoxins. Endotoxin is part of the gram-negative outer membrane LPS. It gets released when the bacteria are damaged and causes widespread inflammation, often involved in sepsis.
And exotoxins.
Those are proteins actively secreted by living bacteria, often with very specific targets. Our source lists great examples in box 6.3c, difficile toxin causing diarrhea, botulinum toxin causing paralysis, cholera toxin causing massive fluid loss,
Diphtheria toxin, tetanus toxin, toxic shock syndrome toxins from Staph aureus, each causing a distinct disease.
powerful and specific weapons.
Another strategy is being an intracellular pathogen, hiding inside our own cells.
Right. Viruses do this, of course, but also bacteria like Salmonella, Listeria, Mycobacterium tuberculosis, even some parasites like Leishmania and fungi like Histoplasma.
And they can even change their gene expression inside the cell, adapting to the environment.
Yeah, responding to stresses like low pH, lack of iron or oxygen makes them very adaptable and hard to eliminate, often leads to chronic infections.
And this adaptability is fueled by genetic diversity.
What's fascinating here is how this diversity boosts pathogenicity. Bacteria can swap genes, especially virulence genes, on plasmids or via phages.
So that idea of the bacterial supergenome, access to a huge pool of potential genes.
Exactly. Viruses do it too, through rapid replication and swapping genetic material. When a strain picks up a really effective combination of virulence genes.
That's when you get epidemic strains, like with new flu pandemics.
Precisely. It's evolution happening right before our eyes, often with devastating consequences.
So the pathogen has all these weapons. What about the host response? Our defenses.
Our innate and adaptive immune systems. They're crucial for controlling normal flora and fighting off pathogens. We covered the basics in a previous deep dive.
But here the focus is on how that response itself contributes to the disease.
It's that double-edged sword idea again. The harm caused by an infection isn't just the bug's virulence, it's also our response to it.
Sometimes the response is too strong.
Exactly. An excessive immune or inflammatory response can cause significant tissue damage. Think of the cytokines and other factors released that can injure our own cells. Extreme inflammation can lead to shock, organ dysfunction like in sepsis.
And that phenomenon, iris immune reconstitution inflammatory syndrome.
Right. You see it when someone's immune system recovers, like after starting HIV treatment or recovering from neutropenia. Suddenly, the immune system reacts fiercely to previously hidden infections, causing a paradoxical worsening. It's the response, not the bug itself, causing the flare-up. A dysregulated, exuberant response.
A very tricky balance. And one very common response is the febrile response. Fever.
Right. Infection alters our thermoregulation, usually causing a hyperthermia, fever, but sometimes hypothermia too.
And fever is mediated by cytokines, IL-1, IL-6, TNF-alpha.
Yes, pyrogenic cytokines. They're released in response to microbial triggers like LPS or signals from our own damaged cells. They ultimately cause the hypothalamus, our thermostat, to raise the body's set point temperature via prostaglandin E2.
Which leads to things like Rickers, that uncontrollable shivering.
Exactly. That's the body trying to generate heat to reach that new higher set point. Chills, shivering, then often fever and sweating.
And the idea is that the higher temperature actually helps fight the infection.
That's the hypothesis, yeah. Raised body temperature might interfere with how well pathogens replicate or use their virulence factors. Some evidence supports this beneficial role.
But hypothermia in infection, that's less understood.
Much less so.
Yeah.
This raises an important question. Why is infection-driven hypothermia so poorly understood? We know it can happen, often in severe sepsis, and it's generally associated with worse outcomes. But the mechanisms and whether it has any protective role, that's still a major area for research. Definitely needs further study.
Okay, so infection is suspected. How do doctors figure out what's really going on? It's fascinating detective work, a mix of clinical skill and lab science.
Absolutely. The aims are clear. Confirm if there is an infection, identify the specific culprit or culprits, and, crucially, figure out what antimicrobials will work against it to guide treatment.
Often the first clues are things like raised inflammatory markers, like C-reactive protein, or changes in white blood cell counts, right? Responses to the pathogen.
Yes, those non-specific indicators. But then you need more specific tests, which fall into direct or indirect detection methods.
And getting good samples is paramount.
Cannot stress that enough. Careful sampling drastically improves diagnostic yield. And interpreting results, especially cultures, always needs context. Knowing the normal flora of the site is essential to avoid misdiagnosis.
Plus, test performance sensitivity specificity isn't fixed. It varies with timing, how common the disease is.
Which is why that communication loop between the clinician at the bedside and the microbiologist in the lab is absolutely vital. Constant dialogue ensures tests are chosen wisely and results interpreted correctly.
Okay, let's look at direct detection first, finding the pathogen itself or its components. These methods are great because they can be rapid, work for unculturable bugs, and sometimes hint at sensitivity.
Starting with seeing the whole organism, microscopy.
Right. Bright field microscopy is the standard, often using stains. Gram stain is the classic for bacteria.
Positive or negative shape.
Seal-Nielsen, or auramine, for TB bacilli.
Okay.
Looking for those acid-fast bugs. And in tissue samples, histology stains show the bug and the damage it's causing.
Then there's dark field microscopy. Makes things appear bright against black. Good for taking thin spirochetes like in syphilis.
Electron microscopy can see viruses in stool or fluids, but
Mostly replaced by molecular tests now. It's cumbersome.
and flow cytometry can analyze particles in fluids like urine, detecting bacteria based on size and light scatter. But it can sometimes be fooled by other particles. Needs careful interpretation.
So seeing the whole bug is one way. The other is detecting its components.
Like nucleic acid amplification tests, NATs. These are incredibly sensitive and relatively fast.
PCR is the most famous example.
Exactly, polymerase chain reaction. You find a specific DNA or RNA sequence using primers, then amplify it exponentially. For RNA viruses like HCV or HIV, you use RT-PCR reverse transcription first.
And modern versions are often real-time, using fluorescence to quantify the amount.
Plus, multiplex PCR lets you look for several different pathogens simultaneously from one sample. Very efficient.
The applications are huge. Sequencing the amplified DNA can identify specific strains, detect resistance genes like MECA for MRSA, or virulence factors.
So knots are used everywhere now. Virology, bacteriology for tricky samples like CSF or tissue, even mycology and parasitology. Especially good for those bugs we can't culture, like Trophroma whipli.
And you can also detect other components, like antigens and toxins.
Right, like the Legionella antigen in urine or cryptococcal antigen in CSF. These rely on specific antibody-antigen binding reactions, or detecting the C. difficile toxin directly in stool, sometimes more relevant than finding the bacteria itself.
And this really highlights that crucial shift in diagnostics we talked about. It's not just about seeing or growing the bug anymore. It's about identifying its unique genetic signature, its specific proteins or toxins.
Giving us faster, more precise answers.
Exactly. NATs, antigen tests. They offer sensitivity and speed, allowing clinicians to make informed decisions much earlier, sometimes before culture results are even back. It's a huge leap.
But culture still has its place, the traditional workhorse.
Absolutely. Growing bacteria and fungi from samples on artificial media, it's still essential. It confirms the pathogen is viable, allows detailed identification, susceptibility testing, and even subtyping for tracking outbreaks.
Ex vivo culture in cells, mostly for viruses, is less common now due to native appearance.
Right. But in vitro culture for bacteria and fungi remains vital.
Though it has limitations. It's not instant, takes hours, days, even weeks for slow growers like M. tuberculosis.
And a negative culture doesn't definitively rule out infection. The bug might be there in low numbers or just not grow in the lab conditions.
Or it might be uncultivable like M. leprae.
Exactly.
A classic example of culture's importance is blood culture, detecting bacteria or fungi in the bloodstream, bacteremia or fungemia.
Critical for diagnosing bloodstream infections, you take blood, put it into special liquid media, and incubate it in automated systems that monitor for growth, often by detecting CO2 production.
If it flags positive, you gram stain it, do further tests, increasingly using MALDI-TOF for rapid ID.
And taking multiple sets helps distinguish real, persistent infection from transient bugs or contamination. That flowchart in Figure 6.6 lays out the whole process.
And you mentioned the importance of preliminary results.
Hugely important. That initial gram stain results from a positive blood culture, maybe some preliminary sensitivities. Getting that info to the clinician quickly is often more critical for immediate patient care than the final detailed report that comes later.
It guides those crucial early antibiotic choices.
Precisely. It's about actionable information, fast. That clinician-lab communication again.
Okay, so that's direct detection. What if we can't find the bug directly? We look for the body's response, indirect detection.
Primarily looking for antibodies. Serology.
Detecting the host's immune response, usually antibodies in the blood. Useful when the bug is hard to find or gone already.
Results often given as titers, that dilution factor.
And diagnosis often hinges on seroconversion, going from negative to positive, or seeing that fourfold rise in titer between acute and convalescent samples.
Or sometimes detecting IgM antibodies, which appear earlier in the infection.
But limitations exist. Needs a working immune system, so can be unreliable in immunocompromised patients.
And often gives a retrospective diagnosis telling you about a past infection unless it's IgM or a chronic infection.
You mentioned those windows of opportunity before? Figure 6.8?
Yes, what's fascinating is how the timing varies. Nucleic acid might be detectable first, then antigen, then IgM, then long-lasting IgG. You have to pick the right test for the right time window based on when the patient got sick.
Strategic testing. So what are the main antibody-antigen detection methods?
ELISA enzyme-linked immunosorbent assay is a big one. Uses enzyme-linked antibodies to create a color change if the target antigen or antibody is present. Very versatile.
Then immunoblotting, or Western blot. Separates proteins by size, transfers them to a membrane, then uses patient serum and labeled antibodies to detect specific binding. Highly specific, often used for confirmation.
Like for Lyme disease.
Immunofluorescence assays, IFAs, use fluorescent labels, either detecting patient antibodies binding to known antigens on a slide, or using labeled antibodies to detect the organism directly in samples, viewed under a special microscope.
The complement fixation test, CFT, is older. It measures how much complement gets used up when patient antibodies bind to a test antigen. Less common now.
Agglutination tests are based on visible clumping, either direct, patient serum plus organism suspension, or indirect, antigen-coated particles plus serum, or reverse passive, antibody-coated particles plus sample to detect antigen.
Those historical tests like Weil, Felix, and Whittle fall here, still used sometimes despite limitations.
Immunodiffusion involves antigens and antibodies migrating through gel, forming visible precipitin lines, used for some fungal diagnostics.
And immunochromatography, those rapid lateral flow tests, like pregnancy tests but for infectious antigens. A line appears if the antigen is present. Great for point of care.
Like rapid HIV or malaria tests.
Exactly. Quick, easy, field deployable.
Finally, under indirect methods, there are antibody-independent specific immunological tests.
The main one being the interferon gamma release assay, or IGRA, mostly used for latent TB infection.
It measures T-cell response to specific TB antigens. But crucially, it can't distinguish between latent and active TB disease.
Right, so its best use is really in low-incidence settings for screening for latent infection, not for diagnosing active disease.
Okay, we've identified the culprit. Now, the million-dollar question. What drug will actually kill it? That's antimicrobial susceptibility testing.
Absolutely crucial for guiding therapy. The fundamentals are straightforward. If an antimicrobial stops the bug growing in vitro, it's susceptible.
We distinguish between bacteriostatic agents. They just inhibit growth reversibly.
And bactericidal agents, they actively kill the cells. Similar terms apply to fungi and viruses.
Key metrics are the MIC, minimum inhibitory concentration, the lowest drug level to stop growth.
And the MBC, minimum bactericidal concentration, the lowest level to actually kill.
And labs use breakpoints. These are preset MIC thresholds to classify an organism as susceptible, intermediate, or resistant based on expected drug levels in the body and clinical outcomes.
But this leads to that important question we touched on. Why doesn't susceptibility in the lab, in vitro, guarantee success in the patient, in vivo?
Right, it seems like it should, but
It's much more complex. Clinical response depends on so many other things. The patient's immune system, how the drug actually gets absorbed and distributed in their body, pharmacokinetics, other illnesses they have, the specific dose used, all interact with the MIC-MBC.
So the lab result is a vital guide, massively increasing the chance of success.
But it's not an absolute guarantee. Clinical judgment remains essential.
How is testing actually done? What are the methods?
The classic is disc diffusion. You spread bacteria on an agar plate, place antibiotic-impregnated discs on top. The drug diffuses out, creating a gradient.
And where the concentration is above the MIC, you get a clear zone where bacteria can't grow the zone of inhibition.
Right. And the size of that zone relates to the MIC. Bigger zone, more susceptible. Similar idea with diffusion strips. They have a built-in concentration gradient, and you read the MIC directly where the edge of the zone intersects the strip. Practical, widely used methods.
Okay, zooming out from the individual patient now. Understanding how infections move through populations, that's epidemiology, and it's critical for public health.
Because these diseases are often communicable, a diagnosis in one person immediately raises questions about exposure in others. Might need isolation, might trigger an outbreak investigation.
Let's look at geographical and temporal patterns. Some diseases are endemic.
Meaning constantly present in a particular area or population, often tied to a restricted reservoir, vector, or specific environmental needs, or maybe limited by geography or vaccination coverage.
But these patterns can change, right?
Absolutely. Animal reservoirs expand, think Lyme disease spreading with deer populations. Vectors travel airport malaria. Host ranges extend schistosomiasis after dam building. Human migration spreads resistant bugs like CPE.
Public health breakdowns can let things like diphtheria resurface.
Yeah.
And climate change.
Big factor now. Affecting the range of vector-borne diseases like dengue, Rift Valley fever, the map is constantly being redrawn.
Then we have emerging and re-emerging diseases.
Emerging means newly appeared or rapidly increasing in range or numbers. Re-emerging means something we thought was controlled is bouncing back.
Many emerge from animals, adapting to infect humans. Zoonotic spillover.
Exactly. HIV from primates is the classic example. Figure 6.11 lists many others: Ebola, Zika, MERS-CoV, anthrax, a constant stream of new or returning threats.
Where did these pathogens hang out? The reservoirs of infection.
The CDC definition is good, where the agent normally lives, replicates, and depends for survival before jumping to a host.
Can be human reservoirs, people who are colonized or actively infected.
Like with Staph aureus, Strep pyogenes, C. difficile. For ongoing transmission, the infection often needs to be long-lasting, like TB or STIs, and some, like measles, only have human reservoirs.
Or animal reservoirs for zoonoses. Diseases naturally pass from vertebrate animals to humans.
And the animals might not even look sick. Transmission can happen in many ways, and sometimes that initial animal-to-human jump leads to secondary human-to-human spread. Q fever, brucellosis, Ebola.
And environmental reservoirs, soil, water.
Yes, pathogens acquired directly from the environment, though often the environment is more like a temporary holding place or conduit, with the pathogen mainly maintained in animal or human reservoirs.
Okay, so the reservoir is the source. How does it get to the host? Transmission of infection.
Multiple routes: respiratory inhalation, fecal oral ingestion, sexually transmitted direct mucous membrane contact, blood-borne direct inoculation.
Direct contact usually needs broken skin, like a wound.
Right, and then indirect via vector or fomite. Vectors are living carriers, mosquitoes, fleas. Fomites are inanimate objects, doorknobs, phones, medical equipment, especially important for HCAIs.
And whether infection happens depends on the bug's virulence and the host's susceptibility. We also need to understand timing, the incubation period.
Time between exposure and first symptoms. Hugely variable hours for some things, years for others, like rabies. Box 6.6 gives examples.
And the period of infectivity, when the person can actually spread it to others.
Also variable and might start before symptoms appear. Box 6.7 has examples like chickenpox, measles.
Why is knowing these periods so vital, even if they're imprecise?
Because it's the foundation of control. It tells you when to isolate someone, how long for, and who needs to be traced as a contact. Even approximate windows allow public health to act to break those transmission chains. It's absolutely fundamental for outbreak management.
And then there's the darker side, deliberate release.
Bioterrorism.
Using pathogens as weapons. It's happened salmonella in salad bars, anthrax spores in the mail.
High-risk agents include anthrax, plague, smallpox, botulism toxin.
It just underscores that dual-use nature of biology. The knowledge that heals can also harm, requires constant vigilance, rapid detection, international cooperation. A sobering reality. Okay, knowing the enemy and how it spreads. How do we systematically fight back? That's infection prevention and control, IPC.
It's the whole suite of measures used to break that chain of infection we talked about, our collective defense strategy.
A huge focus area is healthcare-associated infection, HCAI. Infections picked up in hospitals or clinics.
The risk is significant, maybe 10% in developed countries, and the big worry is multi-resistant bacteria, MRSA, ESBLs, C. difficile, GRE.
Plus C. difficile norovirus, things that spread easily in healthcare settings. Figure 6.12 shows common examples like catheter infections, surgical site infections.
And it really connects the micro and the macro, doesn't it? The pathogen biology meets the healthcare system complexity. It's a constant battle in hospitals against adapting bugs and vulnerable patients, requires tireless effort.
So what are the actual IPC measures? Box 6.8 lists a whole range.
They operate at multiple levels at the institutional level. Proper waste disposal, managing spills, cleaning equipment in the environment, specialized ventilation, sterilization, food hygiene, laundry.
For healthcare staff, education, hand hygiene, crucial. Sharps, safety, PPE use, masks, gloves, gowns, staff screening, immunizations, post-exposure plans.
In day-to-day clinical practice, smart antibiotic use, stewardship, aseptic technique for procedures, preventative antibiotics before surgery, patient screening for key resistant bugs.
And then the response to infections. Surveillance for outbreaks, using prophylactic antibiotics for contacts, isolation, controlling reservoirs and vectors. It's a comprehensive toolkit.
Layer upon layer of defense.
And you highlighted hand hygiene. Seems basic, but maybe the most important. Figure 6.13.
Absolutely fundamental. Mandatory before and after every patient contact. Alcohol gel is usually fine.
Unless hands are visibly dirty or after more intense contact and washed with soap and water.
And figure 6.13 shows the technique getting fingertips, web spaces, backs of hands for a good 40-60 seconds. Small action, huge impact.
Then sometimes you need isolation precautions, box 6.9, extra measures. For airborne bugs like TB or measles, negative pressure rooms, N95 masks for staff.
For contact spread like C. diff or MRSA, private room ideally, gloves and gowns for staff.
For droplets spread like flu or meningitis, private room or space, surgical masks for close contact staff.
Some infections like smallpox need multiple types of precautions. And always, strict aseptic technique for any invasive procedure.
What happens when these measures fail or a new threat emerges? Outbreaks of infection.
Understanding the terminology is key. Box 6.0. A cluster is just cases grouped together. An epidemic or outbreak is more cases than expected. A pandemic is a global epidemic.
We track the index case, first one found, primary cases from the source, secondary cases from primary cases.
Outbreaks can be common source, ongoing exposure, point source, single exposure event, or spread person to person.
Confirming an outbreak often involves typing the organisms to ensure they're identical. If not, it's a pseudo-outbreak.
Investigation involves defining cases, finding them, plotting the timeline, outbreak curve, gathering demographics, maybe doing a case control study to pinpoint the source.
Management is about control and communication, and surveillance is key for prevention and early detection.
Hospitals track alert organisms. Nationally, there are notifiable diseases, box 6.11, that must be reported for reasons ranging from potential spread, import risk, public health failures, food safety, vaccine issues, new threats, or bioterrorism concerns.
It really demonstrates that layered approach from the patient bedside right up to global public health networks.
Totally interconnected. Individual diagnosis informs population surveillance, which in turn informs individual prevention and control strategies. It's a constant cycle of information and action.
Okay, let's shift to a more proactive strategy, one of medicine's greatest successes, immunization, preventing disease before it starts.
A cornerstone of public health. Two main types, passive and active.
Passive immunization is giving preformed antibodies, right? Usually from blood products.
Exactly. Protection is immediate but temporary, weeks or months, used mainly for post-exposure protection or prevention in specific high-risk situations. Box 6.12 lists examples like immunoglobulin for hepatitis A or B exposure, tetanus, rabies, chickenpox contacts.
But the preferred approach for long-term protection is active immunization, vaccination.
Stimulating the body's own immune system to create lasting defenses by exposing it to whole organisms or parts of organisms. Box 6.13.
What are the main types of vaccine?
Broadly, whole cell or component vaccines. Whole cell vaccines use the entire organism. Can be live attenuated, weakened, but still alive.
Like MMR, oral polio, BCG.
Right. They replicate a bit, give a strong immune response, T-cell and antibody, but generally not for the severely immunocompromised due to the live nature. Or they can be inactivated, killed, whole cell vaccines like for Hep A, injected polio, IPV, rabies.
Then component vaccines use just pieces of the pathogen.
Like polysaccharide-only vaccines, example, PPV for pneumococcus. These often give weaker, shorter-lived antibody responses without good T-cell help or memory.
But the big advance was conjugate vaccines, linking the polysaccharide to a protein carrier.
Yes. Like Hib, the newer pneumococcal conjugate vaccine, PCV. This recruits T-cell help, giving a much stronger, longer-lasting response with immunological memory. A huge improvement, especially for infants.
We also have toxoids, inactivated toxins, like for diphtheria and tetanus.
And often adjuvants, like aluminum salts, are added to boost the immune response to the vaccine components.
Why are those conjugate vaccines so much better for memory? What's the immunology?
It's about engaging T helper cells. Polysaccharides alone are T-independent antigens. They stimulate B cells directly but don't generate good memory. By linking the polysaccharide to a protein, the immune system processes it like a protein antigen, involving T helper cells. These T cells then provide signals to the B cells for a much more robust response, more antibody, class switching, and crucially, the generation of long-lived memory B and T cells. That's the key to durable immunity. Different vaccines harness different arms of the immune system.
Clever. How are vaccines used?
Can be universal programs for whole populations or targeted to high-risk groups, travel, occupation, health conditions. Ring vaccination is used in outbreaks vaccinating contacts around a case to stop spread.
Primary goal is preventing the disease, but sometimes there are other benefits.
Like the HPV vaccine preventing cancer, a major secondary benefit. Box 6.14 covers individual considerations contraindications, like severe allergy, or for live vaccines, pregnancy, severe immunosuppression, spacing, interactions. But common chronic things like asthma usually aren't a reason not to vaccinate.
And the success of vaccination can be profound. Herd immunity.
Right. When enough people are immune, the virus or bacteria can't find enough susceptible hosts to keep spreading. It protects the unvaccinated, too.
Leading even to eradication in some cases. Smallpox gone.
Declared eradicated in 1980, polio dramatically reduced since the WHO eradication goal in 1988, down from hundreds of thousands of cases to double digits annually now. Schedules vary, catch-up is possible.
It really showcases what global public health can achieve. Truly monumental.
Absolutely. Smallpox eradication is arguably one of humanity's greatest public health triumphs. It shows the power of coordinated, sustained vaccination efforts and offers hope for tackling other major infectious diseases globally, a real beacon of success.
So vaccines prevent, antimicrobials treat. But our heavy reliance on antimicrobials has fueled a massive, growing problem, resistance. This brings us to antimicrobial stewardship, AMS.
Basically, the organized effort to use antibiotics wisely. Systems and processes within a hospital, region, or country to optimize antimicrobial use.
The goals being better patient outcomes and slowing down the development of antimicrobial resistance, AMR.
And AMS and infection control, IPC, are totally linked. Figure 6.14. Good stewardship reduces infections. Good IPC reduces the need for antibiotics. They work together.
AMS programs involve things like treatment guidelines, restricted formularies, specialist input.
Exactly. Trying to ensure the right drug, right dose, right duration, only when needed.
But why is AMR inevitable? Why does resistance develop?
Well, microorganisms have been dealing with naturally produced antibiotics in the environment for millions of years. They've co-evolved resistance mechanisms. It's natural selection.
So it's not new, but we've accelerated it massively.
Precisely. We distinguish intrinsic resistance. A bug is just naturally resistant from acquired resistance. Acquired resistance comes from random mutations or, much more commonly, getting resistance genes from other bacteria.
Often on those plasmids, right? Horizontal gene transfer?
Yes. Plasmids carrying multiple resistance genes can spread very efficiently through bacterial populations. That's a huge driver of the problem.
And the mechanisms of resistance are diverse. Figure 6.16. Bacteria can become impermeable, block the drug from getting in.
Or they can pump it back out, active efflux.
Or they can change the drug's target inside the cell so the drug can't bind anymore.
Like MRSA changing its penicillin-binding protein. Or resistance to rifampicin or quinolones via target mutations.
And maybe the most famous, bacteria producing enzymes that just chew up the antibiotic.
Like penicillinases destroying penicillin, or ESBLs and carbapinemases destroying broader spectrum beta-lactams. Very effective defense mechanisms.
What factors promote resistance?
Obvious ones are inappropriate use antibiotics for viruses, for example. Incorrect dosing, unnecessarily long courses, using them in agriculture as growth promoters.
But crucially, any antibiotic use creates selection pressure. Even perfectly appropriate use favors the survival and growth of any resistant bacteria that happen to be present.
Exactly. Using combinations might slow resistance in the target bug, but doesn't stop it emerging in the bystander normal flora. We see the results everywhere. MDR-TB, XDR-TB resistance rates climbing globally.
People talk about the post-antibiotic era. Is that realistic?
Maybe not a sudden cliff edge, but certainly a gradual, relentless erosion of effectiveness. We're forced to use older, more toxic drugs or newer, more expensive ones, or sometimes we just run out of options. It raises that critical question: what does it mean for you, for society, when these miracle drugs stop working reliably? It affects surgery, cancer care, organ transplants, everything. A fundamental threat to modern medicine.
Okay, let's get practical.
Yeah.
How do we actually treat infections with drugs? It's more than just picking an antibiotic.
Absolutely. Key components of treatment involve optimizing the antimicrobial therapy itself, choosing wisely, minimizing resistance pressure, limiting collateral damage to good bacteria.
But also addressing underlying issues, right? Controlling blood sugar and diabetes, managing HIV viral load.
Yes, treating the whole patient.
Yeah.
And considering adjuvant therapy, maybe removing an infected catheter, draining an abscess, removing dead tissue, things the antibiotics alone can't fix.
Managing complications like sepsis or kidney injury.
Yeah.
And if it's communicable, thinking about contacts, isolation, prophylaxis, vaccination.
It's a holistic approach. Now, the principles of antimicrobial therapy. Sometimes you need to treat empirically, fast, like in suspected meningitis, before you know the exact cause.
Other times, you can wait for confirmation. The choice depends on the likely bugs, local resistance, guidelines.
And understanding antimicrobial action and spectrum is key. How does the drug work? What range of bugs does it cover? Box 6.15 and 6.16 detail this for major classes and common bacteria.
Generally prefer bactericidal killing agents over bacteriostatic inhibiting ones in severe infections or immunocompromised hosts.
Yes, you want a more definitive effect then.
We talked about empiric versus targeted therapy, starting broad based on likelihoods, then narrowing down once you have the specific bug and its sensitivities.
The start smart, then focus principle, figure 6.15, essential for stewardship. Empiric choices are guided by site, patient factors, and crucial local resistance data.
When is combination therapy used? Multiple drugs together.
Several reasons. To boost effectiveness, maybe against biofilms, when you need broad coverage for mixed infections. Or, critically, to prevent resistance developing, like in TB or HIV treatment, makes it harder for the bug to become resistant to everything at once.
And duration of therapy, box 6.17. How long do you treat?
Varies hugely. Depends on severity, site access. Often limited hard evidence for exact durations. Switch from IV to oral when possible. General principle. Stop when the infection is clinically resolved. Examples range from single-dose gonorrhea to months TB, or even longer, some fungal infections.
Now for the really detailed science. Pharmacokinetics, PK, and pharmacodynamics, PD. How the drug behaves in the body and how it affects the bug.
Right. PK is what the body does to the drug absorption, distribution, metabolism, excretion. Does enough drug actually get to the site of infection?
Like IV might be needed if gut absorption is poor in sepsis, or some drugs don't get into the brain well unless there's inflammation.
Exactly. And for a UTI, you want a drug excreted in the urine. PD is what the drug does to the bug, the relationship between concentration and killing effect. Figure 6.17.
We look at Cmax, peak level, AUC, total exposure, relative to the MIC.
Some drugs have concentration-dependent killing, like aminoglycosides. The higher the peak, Cmax above the MIC, the better they kill. That's why we give high doses less frequently, like once daily gentamicin. Maximizes the peak, minimizes toxicity.
Others have time-dependent killing, like beta-lactams, vancomycin. Efficacy depends on how long the concentration stays above the MIC.
Which dictates more frequent dosing or even continuous infusions for severe infections to keep levels consistently high enough. Some drugs also have post-antibiotic effects, continuing to work even after levels drop.
This complexity leads to therapeutic drug monitoring, TDM.
Yes, measuring drug levels in the patient. Essential for drugs with a narrow therapeutic window, like aminoglycosides, to avoid toxicity. Also useful for drugs with variable PK, like vancomycin, to ensure levels are adequate. Specific targets depend on the drug and situation, sometimes using tools like the Hartford nomogram for gentamicin, figure 6.18. Finally, using antimicrobials preventatively, antimicrobial prophylaxis.
Primary prophylaxis is before potential exposure, before surgery, after contact with certain infections, or for high-risk patients like post-splenectomy. Secondary prophylaxis is long-term prevention in patients prone to recurrence, like fungal prophylaxis in leukemia patients, or preventing opportunistic infections in HIV until immunity recovers. Box 6.18 lists many examples across different pathogen types.
Okay, let's dive into the specific drugs themselves, mastering the details of the antimicrobial arsenal. But first, some general considerations. Pregnancy is a big one, box 6.19. Huge. Some drugs are absolutely contraindicated. Tetracyclines, quinolones, chloramphenicol. Others relatively contraindicated, aminoglycosides. Many are considered generally safe, penicillin, cephalosporins, erythromycin. Always a risk-benefit calculation.
And old age, box 6.0, brings changes. Higher risk of C. diff, altered kidney function affecting drug clearance and toxicity risk, reduced liver metabolism, increased CNS side effects with some drugs. Needs careful dosing and awareness.
Definitely requires adjustment.
Right, let's tackle A, antibacterial agents, starting with the workhorses. Beta-lactam antibiotics, box 6.21, all share that core ring structure, inhibit cell wall synthesis, target PVPs, and are bactericidal.
They generally distribute well into many tissues, less so into CSF unless inflamed. Generally safe in pregnancy, with a few exceptions. Adverse effects range from common rashes and GI upset to rare severe allergies, blood count changes, kidney issues, seizures at high doses.
Within this group, penicillins, natural ones, benzylpenicillin, mainly for gram-positives. Penicillin-resistant ones, flucloxacillin for Staph aureus, if not MRSA. Amidopenicillins, amoxicillin, add some gram-negative cover, often need a beta-lactamase inhibitor like clavulanate due to resistance. Extended spectrum ones, piperacillin, cover Pseudomonas, especially with tazobactam.
Cephalosporins and cefamycins, box 6.22, are broad spectrum grouped in generations. First gen good for gram-positives, later generations add more gram-negative cover, with fifth gen hitting MRSA and Pseudomonas, ceftobiprole. Associated with C. diff, don't cover enterococci well, except newest ones.
Monobactams, Astrianam, unique, only gram-negative activity, often safe in penicillin allergy. Carbapenems, the big guns. Broadest spectrum beta-lactams cover gram-positives, gram-negatives, anaerobes, reserved for serious resistant infections.
Moving on. Macrolide and lincosamide antibiotics. Macrolides, erythromycin, azithromycin, and clindamycin. Mostly bacteriostatic, inhibit protein synthesis. Good for atypical pneumonias, some STIs, macrolides, skin soft tissue bone infections, clindamycin. Clindamycin has that high risk of C. diff. Macrolides can cause GI upset, rarely heart rhythm issues.
Aminoglycosides. Gentamicin, amicus, primarily for serious gram-negative infections. Synergistic with beta-lactams for endocarditis biofilms. Need IVIM, negligible oral absorption. Risk of kidney or toxicity requires monitoring. Exhibit concentration-dependent killing and post-antibiotic effect. Spectinomycin is related, used for resistant gonorrhea.
Quinolones and fluoroquinolones, ciprofloxacin, levofloxacin, box 6.23, bactericidal broad spectrum, especially fluoroquinolones. Good oral absorption, tissue penetration, used for UTIs, pneumonia, MDR-TB concerns. Tendon rupture, CNS effects, cardiac issues, association with MRSA and C. diff. Resistance is a growing problem, especially with Pseudomonas.
Glycopeptides, vancomycin, teicoplanin. Gram-positive only, crucial for MRSA. Ampicillin-resistant enterococci, given IV except oral vanc for C. diff. Require TDM due to kidney toxicity risk. Lipopeptides, daptomycin, also gram-positive only. MRSA, GRE, IV use, not for pneumonia, can affect muscles. Polymyxins, colistin, back in use for multi-resistant gram-negative CPEs. Significant kidney nerve toxicity risk.
Folate antagonists, trimethoprim sulfonamides, inhibit folate pathway. Cotrimoxazole, TMP-SMX, first line for pneumocystis, also used for staph infections. Dapsone used in leprosy. Risk of skin reactions, blood issues, especially with G6PD deficiency for sulfoniazed dapsone. Oxazolidinones, linezolid, good for resistant gram-positives, MRSA, GRE. Oral RV can cause low blood counts, neuropathy with long use, interact with SSRIs, serotonin syndrome risk.
Other antibacterials, fusidic acid, gram-positive, often combo use. Nitrofurantoin, UTIs only. Fidaxomycin, specific for C. diff. Phosphomycin, UTIs, resistant bugs. Tetracyclines, doxycycline, broad spectrum, good for atypicals, rickettsia. Lyme, resistance common. Bind teeth bones, avoid in kids' pregnancy. Glycosylines, tigecyclines, very broad spectrum, but mortality concerns. Nitroimidazoles, metronidazoles, excellent for anaerobes. Protozoa. Good absorption distribution. Metallic taste, severe reaction with alcohol. Phenocals, chloramphenicol, very broad spectrum, but reserved due to rare but fatal aplastic anemia risk.
Okay, that's a lot of antibacterials. Now for B, antimycobacterial agents, primarily for TB, first line. Isoniazid inhibits mycolic acid, liver toxicity, neuropathy risk. Rifampicin inhibits RNA polymerase, potent enzyme inducer, many drug interactions, orange secretions. Pyrazinamide, mechanism less clear, active in acidic conditions, liver toxicity. Ethambutol inhibits cell wall optic neuritis risk. Streptomycin, aminoglycoside. MDR-SD RTBs, second-line agents are crucial. Fluoroquinolones, other aminoglycosides, newer drugs like daqualine, delaminid. Complex regimens, significant side effects.
Now C, antifungal agents, box 6.24, azoles, fluconazole, voriconazole, itraconazole. Inhibit ergosterol synthesis. Broad use, but drug interactions via P450 enzymes. Side effects include liver issues. Echinocandins, caspofungin. Inhibit cell wall, beta-glucan synthesis. Good for candida, aspergillus, fewer side effects, flebby-only. Polyanes, amphotericin B. Bind cortisol, damage membrane. Very broad spectrum, but conventional form is toxic, kidney damage. Lipid formulations are much safer preferred now. Nystatin is topical only. Other antifungals. Glucytosine, yeast, combo use only. Griseofulvin, skin fungi, largely replaced. Terbinafine, skin nails, rare liver toxicity.
D. Antiviral agents, box 6.25. Many viral infections don't need treatment in healthy people. Antiherpes virus agents, aciclovir and prodrug valacyclovir for HSV-VZV, cantisclovir for CMV, cidofovir, foscarnet for resistant strains, all target viral DNA replication, anti-influenza agents, neuraminidase inhibitors, oseltamivir, zanamivir, shorten illness if given early, useful for high-risk groups during outbreaks. Older drugs, amantadine, largely useless now due to resistance. Other antivirals, ribavirin, HCV, RSV, LESA, specific agents for hepatitis B, C covered elsewhere.
Finally, E, antiparasitic agents. Huge diversity here. Antimalarial agents. Artemisinin derivatives. Artesunate, arquinol, especially for resistant malaria. Adavlaquan proguanil combo. Quinoline compounds, chloroquine, quinine, mefloquine, have historical importance but resistant side effects. Neuropsychiatric with mefloquine limit use. Primaquine for liver stages. Drugs for trypanosomiasis, sleeping sickness, chagas, benznidazole, nifurtimox for chagas, aflornithine, melarsoprol, toxic arsenical, pentamidine, suramin for African forms, often toxic complex administration, other antiprotozoals, antimonials for leishmaniasis, cardiotoxic, agents for amoebiasis, giardiasis like metronidazole, paramomycin, nitazoxanide, drugs against helminths, worms, benzimidazoles, bendazole, are broad spectrum. Praziquantel crucial for schistosomiasis, tapeworms, ivermectin for river blindness, strongyloids, dialocarbamazine for filariasis, many targeted agents reflecting the diversity of parasitic worms. #outro. So what does this all mean? We've just covered a massive amount of ground, really diving deep into the core principles of infectious disease, for the microbes themselves and how they operate.
How we diagnose them, the global strategies for prevention like vaccination, and that incredibly complex field of antimicrobial therapy and resistance.
It really hammers home the complexity, the interconnectedness of it all. To truly master this, you need to understand all these pieces, from molecular mechanisms right up to public health policy.
Absolutely. We've seen how tiny agents have huge global impacts, how our bodies fight back in intricate ways, and how human ingenuity is constantly racing to keep up, especially with challenges like AMR.
If we connect this to the bigger picture,
It's clear this field is never static. Microbes evolve, environments change, human behavior shifts. It demands constant learning, constant critical thinking.
so here's a provocative thought to leave you with. Considering this dynamic nature, pathogens evolving, climate changing, global travel increasing, how do you think the landscape of emerging and re-emerging diseases will shift in the next 10 years? And maybe more importantly, what role will our own behavior individually and collectively play in shaping how vulnerable we are?
A crucial question for our future.
Definitely something to mull over. We hope this deep dive has given you a solid, comprehensive foundation. Whether you use this knowledge professionally, for your own health, or just to better understand the world, we hope it's been valuable. Keep exploring, keep asking questions. Thanks for joining us.
Thank you.
Until next time, keep digging deeper.